Okay, we're live. We are live now. Starting this lecture. It's a very interesting lecture. This is about immunity, pathology. So, immunity is a defense mechanism of the body against infections. The immune system is a collection of cells and molecules that are responsible for defending us against pathogenic microbes and environments. Immune system, there is adaptive and innate immune system. This lecture is important in the educational system. Now, innate immunity, also called natural or native immunity. It's front line of defense. Not specific, no immunologic memory, does not get stronger with more exposures. First immunity is called innate immunity. With innate immunity, we have, it's also called natural immunity. This kind of immunity is native immunity. It is the front line of defense, not specific, not immunological, but it does not get stronger with more time. So we have this live streaming. It's very important live streaming for the people to understand it, and very useful in the process of educational system in order to understand the pathology, specifically the immune system. The first line of defense, we have the first line of defense and the second line of defense. First, physical mechanical barriers, skin, mucus, coughing, sneezing, and urination. Two, chemical barriers, acid secretions, sweat, tears, semen, vaginal secretions, respiratory enzymes, salivary enzymes, breast milks, and breast milk enzymes. The two most important cellular reactions of innate immune state are inflammation and antiviral defense. This is here, I'm posting it right now with my mouse. Down here is very important two most important cellular reactions of innate immune system or immune rate, inflammation and antiviral defense. We should be looking to the stream right now. Yes, this is the mouse. Okay, everything is working right now. Recording has started again. The recording is being rebooted. And this time we're talking about adaptive immunity. Okay, it should be on right away. Very interesting. This slide is talking about adaptive immunity, also called acquired or specific immunity, consists of mechanisms that are stimulated by microbes and are capable of recognizing microbial and non-microbial substances. This adaptive immunity is a specific immunity consists of mechanisms that are stimulated by microbes, capable of recognizing microbial and non-microbial substances. It is adaptive. It is innate. It is not innate immunity. It's the opposite of innate immunity. It does not. The innate immunity does not develop over time, but this one does. That develops over time. This one does. There is adaptive and innate immunity. This one is adaptive. Let's talk a little bit about the features of adaptive immunities. We remember that the subject is pathology and talking about immunology. Features of adaptive immunity, it does develops over time or develops later, more powerful than innate immunity. It consists of lymphocytes and their products. Adaptive immune response is referred to an antigen-specific response. This requires processing a pathogen presenting the Ig2B and T cells, production of antibodies, activation consistent cytokines, T cells, creations of memory cells for long-lasting immunology. This is the adaptive immunity response referred to antigen-specific response. This requires. So we have those 5 requirements for the antigen-specific response of the adaptive immunity. These requirements: number 1, processing the pathogen; number 2, presenting the Ig2B and T cells; number 3, they mean presenting the Ig2, then presenting the antigen to the B and T lymphocytes; number 3, production of antibodies. This is a very important step when they are being produced, the antibodies, or you can say that they are lymphocytes, T, B, and also macrophages and white blood cells. Number 4, activate cytokines or cytotoxins. Now, cytotoxins does cytotoxins with T cells, cytotoxic T cell. Cytotoxic T cell is like T lymphocytes, but it's different because T cells, and it is cytotoxic. Number 5, creation of memory cells for long-lasting immunity. Number 5 is the most important thing. We have it, creation of memory cells for long-lasting immunity. Because memory cells, they do remember the antigen and how to deal with it next time it invades the body. Go to the next slide now. We have differences between innate and adaptive immunity. The main differences between innate and adaptive immunity being that there is, in the innate immunity, there is immediate maximal responses. Immediate maximal response, non-antigen specific. Number 3 is exposure results in no immunological memory. These are the most important specificities of the innate immunity. are the most important differences between innate and adaptive immunity. Let's say that again. Let's revise them and say the innate immunity specificities. The main differences, there are only three. There is immediate maximal response. There is no antigen specific, and they are exposure, and the exposure results in no immunological memory. But when it comes to adaptive immunity, the response is antigen dependent. So it's different with the immediate maximal response and non-antigen specific or the innate immunity, while the adaptive immunity does respond in antigen dependent, not like the innate immunity that is not antigen dependent or not antigen specific. Number two of difference that is mentioned in the adaptive immunity difference from the innate immunity is that there is a lag time between exposure and maximal response. There is a lag time between exposure and maximal response. This lag time being week or 2 weeks for the adaptive immunity, while the innate immunity does not have this lag time. It just starts working after 1 week or 2 weeks of invasion. Number three, it's antigen specific. It depends on the antigen in order to resolve the body and protect it from the antigen that is facing. Number four, exposure results in immunological memory. This is adaptive immunity. We have to be very specific with this adaptive immunity feature. The exposure results in immunological memory. The immunological memory being that the memory cells that remember the pathogen when it invades next time, they will connect with it and will destroy it immediately. Number five, recognition of antibody and T cells receptors. They can recognize the antigen next time they invade the body with the T cell receptors and kill it immediately. Next slide. We have the antigen Ag. The abbreviation of the antigen is Ag. It's defined as a substance, usually protein in nature, which when introduced stimulated antibody production. Antigen is defined as a substance, usually protein in nature. Most antigens are proteins. Some viruses, but they are not protein. They just use the virus mechanism to invade the body. The protein is natural, which when introduced into the tissue stimulates antibody production. Usually the protein in nature stimulates antibodies from the immune system, from the innate and adaptive immune system. Antibody is the opposite of antigen. Antibody AB is a protein substance produced as a result of antigenic stimulation. Circulation antibodies are immunoglobulins IgG, IgA, IgM, IgE, IgD. The antigen may combine with antibody to form antigen antibodies complex. This explains the AB. The AB is a protein substance produced as a result of antigenic stimulation. The AB, we can repeat it just to understand it very well. It is the difference, it's the opposite of AG, antigen, the antibody. It's a protein substance produced as a result of antigenic stimulation. Circulation antibodies are immunoglobulins. So when the antibody just circulates within the body and within the tissues and the circulatory system, being the vein and the muscles of the body, the antibodies are immunoglobulins. They are called immunoglobulins. Immunoglobulins, there are 6 types of immunoglobulins. Actually, just 5. IgG, IgA, IgM, IgE, and IgD. They are the most important ones, of course, being the IgG, starting with the lag phase, and then IgA, then IgM, then IgA, then IgD. Antigen may be combined with antibodies to form antigen-antibody complex. This is the most important part of eliminating the antigen. It's when the antibody or the immunoglobulin antigen combined with antibody and forming antigen-antibody complex. Antigen-antibody complex can be identified by the macrophage and getting macrophages, phagocyte sources, and get rid of the body from the innate response or the adaptive response. Moving to the next slide. This slide is adaptive immunity can classify into two categories. The adaptive immunity can be classified into two categories. First category being the hormonal immunity, second category being the cell-mediated or cellular immunity. Starting with the hormonal immunity. Humoral immunity, which protects against extracellular microbes and their toxins that mediated by the B lymphocytes and their secreted products, antibodies, AB. We will repeat that just to understand it very well for the hormonal immunity. The hormonal immunity protects against extracellular microbes and their toxins that mediated by B lymphocytes and their secreted products, antibodies, AB. Number two, cell-mediated or cellular immunity, which is responsible for the defense against intracellular microbes that mediated by T lymphocytes. T-mediated or cellular, the cell-mediated immunity, defense against intracellular microbes that mediated by T lymphocytes. Hormonal is different from the cell-mediated one which protects against the cellular microbes and their toxins that mediated by B lymphocytes, their secreted products, antibodies, AB. I'll repeat that briefly. Hormonal immunity, which protects against extracellular microbes and their toxins that mediated by the B lymphocytes and their secreted products, antibodies, AB. And then we will repeat the cell-mediated or cellular immunity. And there is a picture explaining the process of light chain and variable region, also the Fab region, also the FR region. Then we have the heavy chain and light chain. What's the difference between heavy and light chain? Being the light chain does have two colors, but the heavy chain has one. Variable region is the interchangeable, and the Fab region doesn't goes just below the variable region. The FR region, it is unicolored region. Humoral immunity, which protects against extracellular microbes and their toxins. We need to understand this humoral immunity in the next slide so that it will be easier to understand it briefly. Protects against extracellular microbes and their toxins are mediated by B lymphocytes and their secreted products, antibodies, AB, antigen, AG. Just to remember. Now, number two of the adaptive immunity can be classified into two categories. The cell-mediated or cellular immunity, which is responsible for the defense against intracellular microbes that mediated by T lymphocytes. and the humoral immunity, which is protecting against extracellular microbes. So the main difference between humoral and cell-mediated or cellular immunity being that humoral is defended or mediated or protects the body against the humoral immunity because it's a humoral, it's protected from the extracellular microbes, and because the other type of adaptive immunity called cellular or cell-mediated immunity is protected from response against intracellular microbes. Intracellular is different from extracellular, as extracellular is from humoral immunity, intracellular is from cell-mediated immunity. Last time we repeat this, adaptive immunity can be classified into two categories, humoral immunity and cell-mediated immunity. Humoral, which protects against extracellular microbes and their toxins that are mediated by B lymphocytes, and their secretory products, antibodies, AB. The cell-mediated, which is responsible for defense against intracellular microbes, mediated by T lymphocytes, not the antibody ABs. What will be the difference, the main two differences between humoral and cell-mediated immunity? The humoral is specific and protects the body against extracellular microbes, and it is mediated, or the toxin is mediated by the B lymphocytes and their secretory products, antibodies AB. So AB and B lymphocytes is different from the T lymphocyte or cell-mediated immunity. Because cell-mediated immunity protects the body from intracellular microbes, and also that is mediated by T lymphocytes. I repeat, T lymphocytes as in T cells. Cytotoxins, T cells. Also there is inside the cell-mediated, the first one being the humoral immunity, which is protection against extracellular antibody or B lymphocytes. Let's move now to the next slide. The cells of adaptive immunity, we have two types of cells that are specific to the adaptive immunity. We just talked about the B and T cells. B that was being the type of antigen that is mediated the body and protects from extracellular microbes, which was within the humoral defense mechanism, while the other type of cellular defense or cell-mediated defense mechanism was about the T lymphocytes protecting the body from inside or intracellular microbes. The cells of adaptive immunity, the number 1 and number 2, starting with number 1. B cells involve in humoral immunity. It's differentiated into plasma cells, which produce antibodies for the specific antigen, and memory cells to provide immunological memory. Let's repeat that briefly, because we are studying this pathological functionality and mechanism. Number 1, B cells involve the humoral immunity, as we mentioned before, and it's differentiated into plasma cells, which produce antibodies for the specific antigen, and memory cells provide immunological memory. So the beta cells differentiated into plasma, producing antibodies, and into memory cells, producing immunological memory. Next, number 2 is T cells. These are the cells that were being used in the cell-mediated immunity of adaptive immunity. Types of T cells, of course, they are cell-mediated immunity, not the humoral immunity. They are cell-mediated immunity, as we mentioned before in the previous slide. This type of T cell, cell-mediated immunity types of T cells, this T cell does not go just like the B cell mentioned before that was involved with the humoral immunity. It was actually the humoral immunity, as we remembered, it causes the production of plasma cells that are produced antibodies and immunological memory that was produced by the memory cells. But this T cells has lots of types. They are cell-mediated immunity. T cell or cell-mediated immunity types of T cells. First and second and third. The first one being that CD4 T cells or T helper cells, of which there are two types, Th1 and Th2. This is the cell-mediated immunity type of T cell. This is one type of T cell, a cell-mediated immunity. This is a type of T cell. Then we have CD4 T cells, which is the first type, or helper cells, of which there are two types of Th1 and Th2. Th1 and Th2 T cells. CD4 T cells. T helper cells. T helper 1 and T helper 2 cells. These are the T cells of the cell-mediated immunity. Humoral immunity is different from the first one, the previous one that we mentioned before. The CD4 T cells. CD4 T cells or T helper cells. They use both names interchangeably. CD4 T cell or T helper cells, which T helper cell 1 and T helper cell 2, Th2. There's two types of T cells. Then we have the CD8 T cells, T killer or cytotoxic T cells. They are not Th cells, they are TK or T killer or cytotoxic T cells, CD8, not CD4. This is the second type of T cells. The third type is involved with the cell-mediated immunity that defends the body from the intracellular microbes. The NK, the natural killer cells, provide the link between the innate and adaptive immunity. Natural killers, NK. So we have NK, we have TH, we have CD8, we have CD4, NK, natural killer. All of them are three parts. NK, TH, and TK or CD8. CD4, CD8, and NK. T cells. And then we have B cells, or the humoral immunity, defending the extracellular microbes. And this type is usually mentioned undifferentiated plasma. That's TCL produced antibodies one specific. The produced B cells, there is no two types, they are just producing plasma, antibodies, and producing memory cells. Moving to the next slide, we have the cells of immune system, the cells of the immune system. Phagocyte, macrophage, dendrite, lymphadocyte, all with pictures relating and showing the types of these cells. Starting with the lymphadocytes, cells of immune system. They could be from the adaptive and could be from the innate immune system. Most importantly, the macrophages and macrophagocytes. Dendrites and lymphadocytes are not as important as the first two cells. We'll be enumerating these cells one by one. Lymphadocytes, which are the mediators of adaptive immunity and can be classified into two types, T and B lymphadocytes. We already talked about T and B lymphadocytes. T, we already mentioned that it has T helper 1, T helper 2, CD4, CD8, and T and K, natural killers, T cells from the cellular-mediated immunity. For the humoral immunity was the B T lymphocytes, which was acting from the plasma and acting to make this plasma and memory cells. Two, we have, after mentioning the lymphocytes, which are the mediators of adaptive immunity, and can be classified into two types, T and B lymphocytes. Of course, the innate immunity we didn't mention any cells relating to the innate immunity. We just mentioned cells that are related to the innate, to the adaptive immunity, which is T lymphocytes, B lymphocytes, and their types, of course. And now we are talking about the antigen-presenting cells, APCS, antigen-presenting cells, that capture and display antigen dendritic cells, macrophages, and B cells. Antigen-presenting cells, APCs. What are these cells? These APCs, antigen-presenting cells, capture and display antigen dendritic cells, macrophages, and B cells. Antigen presenting cells, APCs, capture and display antigen dendritic cells, which have been drawn below, phagocytes, macrophages, B cells. So B lymphocytes is actually an antigen presenting cell. There is no drawing showing that cell, but it's mainly a cell that is from the antigen presenting cell of the immune system, probably from the adaptive immunity. The antigen presenting cells, APCs, that capture and display antigen capture and display antigen, which are 1, 2, 3 types of cells: APCs, antigen-presenting cells, dendritic cells, which has been drawn, macrophages, which has been drawn, and B cells. And B cells. We haven't mentioned phagocytes. We just macro and dendritic APCs. These are the cells of immune system. We will be mentioning the third type of immune system, then we'll be taking a brief and short rest, about 5 seconds, 30 seconds, not 5 seconds, 30 seconds. Then we'll be coming back soon. 3 other factors, other effectors, cells such as leukocytes, NK, and plasma cells, natural killers. We already talked about natural killers being one type of T lymphadocytes, not the T helper, not the CD8 T killer. It is NK killer, natural killer, plasma cells. It has not been drawn in this slide, but these are the other factors, other factor cells, such as leukocytes, which is a phagocyte, and also NK natural killer, and plasma cells. Plasma cells. Plasma cells being one of the B cells, actually. As we can see in the previous slide. B cell is the immunity differentiate into plasma cell, which produce antibodies from the specific antigen primarily that cells provide immunity. Here we have humoral cells different into plasma cells. Plasma cells produce antibodies for the specific antigen and memory cells to provide immunological memory. This is plasma cell, and here plasma cell also has been mentioned as type of the cells of immune system from the other cells with the leukocytes, natural killer, and plasma cells. From the other cells of natural killer, leukocytes, plasma cells, not the apex antigen-presenting cells, because antigen-presenting cells are dendrites, macrophages, B cells, and of course the T lymphocytes and B lymphocytes are also important. Now taking a rest for about 1 minute. Okay, sorry about that, everyone, but we are taking another minute of rest. Please wait, and we will be back soon. Okay, sorry about this technical difficulty we were facing, but hopefully now the microphone is working again. This is slide number 9. Cells of the immune system. Just testing the voice. Okay, everything seems to be fine now. Cells of the immune system. Let's repeat these cells of the immune system briefly. Number 1, lymphadocytes. Number 2, antigen-presenting cells. Number 3, other factor cells such as leukocytes, NK, and plasma cells. We have phagocytes and macrophage, dendritic, and lymphadocytes. The phagocytes being with other factors, effector cells. The macrophage and dendritic being with the NAPCS, antigen presenting cells. APCS, antigen presenting cells. Dendrites and macrophages that capture and display antigen. Dendritic cell, macrophage, and B cells are from the APs. Antigen presenting cells, they present antigen. The lymphadocyte being on its own type of cells, we have B and T lymphadocytes, which are mediators of adaptive immunity and can classify into 2 types, T and B lymphadocytes. Moving on to the next slide. Major histocompatibility complexes. Major histocompatibility complexes. MHC. That's actually the first time I'm studying this subject, so bear up with me now and let's try to understand what's happening with the MHC, major histocompatible complex. Major histocompatibility complexes, MHC, are molecules that present antigenic cytolic components to the exterior for the purpose of initiating an immune response. MHC1, which are found on all nucleated cells of the body, MHC2, which are found on the macrophage, dendritic cells, and B cells, antigenic presenting cells, APCs, cytokines, messenger molecules of the immune system. We will repeat this in order to understand it more. Of course, if we have cytokines, the messenger molecule of the immune system, cytokines, is just a messenger of the immune system, adaptive and innate. Now, major histocompatibility complexes, MHCR, molecules that present antigenic, systolic, not cytokines, it's systolic, not diastolic, systolic, major, the antigenic, systolic components to the exterior of the purpose of initiating an immune response. Major compatibility complexes, MHC, major histocompatibility complexes. Molecules represent antigenic, systolic components of the exterior for the purpose of initiating an immune response. Molecules represent antigenic components of the external. So the major importance of the histocompatibility complexes is that they provide, or they are the components that present antigenic, systolic. They are antigen of the pathogen, so they can kill the pathogen quite easily in this location of the major histocompatibility complexes. Systolic components of the exterior purpose of the initiation of an immune response. This is the first major histocompatibility. We are now pointing it with a cursor, with the mouse. Major histocompatibility complex are the molecules that present antigenic, systolic component to the exterior of the purpose of initiating an immune response. They are divided into two types. The major histocompatibility 1 and the major histocompatibility 2. Starting with the one which are found on all nucleated cells of the body. The major histocompatibility 1 found on all nucleated cells of the body. Found on all nucleated cells. Major histocompatibility complex 2, which are found on macrophage, dendritic cells, and B cells, equal antigen presenting cells, APCs. The major histocompatibility complexes 2, which are found on the macrophage, dendritic cells, and B cells. They are APC cells. We mentioned before on the previous slides, APC cells include macrophages and dendritics, which equal to antigen presenting cells, APCs, which are the NHC2 major histocompatibility complexes. Different from major histocompatibility 1, which are found on the non-nucleated cells of the body. Two types of major histocompatibility are molecules representing antigen systolic component to the external of the purpose initiating of immune response. This was slide number 10 out of 37. Moving to the next slide, we have the hypersensitivity. One second, just fixing the mic. The hyper, one second. Just fixing the mic for the better audio. Hypersensitivity, it is an excessive response to an antigen. It is not a response to the pathogen, it's actually to the antigen or a pathogen, just an excessive response. Overall, it's called hypersensitivity. Hypersensitivity disease may be caused by two types of abnormal immune response. Posting it right now with a mouse. We have the two types of abnormal immune response. First, immune response to foreign antigen may be dysregulated or uncontrolled, resulting in tissue injury. This is the first response to foreign antigen. This regulated, uncontrolled, resulting in tissue injury. Moving on to the second hypersensitivity response. It is not responsible for antigen body, that's right, but it's actually responsible for antigen may be regulated or uncontrolled, resulting in tissue injury. The second being that the immune response may be directed against self-antigen, autoantigen. Second, the immune response may be directed against self-antigen, autoantigen. So the first one is a regulated response. The second, directed against self-antigen. This is the slide number 11. The excess response to antigen hypersensitivity may be caused by two types of abnormal immune response. Moving to the next slide, we have the type 1 hypersensitivity, which is uncontrolled, as we mentioned before. The immediate anaphylactic reaction type 1 hypersensitivity is anaphylactic reaction, or is called anaphylactic shock. That is mediated by IgE attached to mast cells and trigger degranulations of chemical mediators. Type 1 hypersensitivity mediated anaphylactic reaction. The mediated by not cellular, actually not humoral. It is type of adaptive immune system because it is directed with the IgE, and the IgE go and attach to mast cells. Then will be IgA, IgB, IgM, and IgG, all within the phase of lag. Moving on with the disease. One second. There was a mistake with the microphone, but now it has been fixed. So there is no need to worry. Now it's better, and you might be able to hear me much better than the previous location. Checking, testing the microphone, 1, 2, 3, check. Testing. Okay, testing. 1, 2, 3, testing. I think it is good now. Let's move back and continue the subject that we were talking about. The type 1 hypersensitivity immediate anaphylactic reaction immediate by IgE attached to mast cells and trigger degranulation of chemical mediators. Type 1 hypersensitivity immediate anaphylactic shock. This type is mediated by IgE, attached to mast cells and trigger degranulation of chemical mediators. Mediated by IgG and attached to mast cells and trigger degranulation of chemical mediators. Mast allergens promote a Th2 cell that secrete IL-4, IL-5 and IL-13, which in turn favor IgE class. Mast allergens promote a Th2, T helper cell 2. The most allergens promote T helper cell 2. These are all actually the type 1 hypersensitivity is mediated by IgE and then promoted by Th1, secrete IL-4, 5 and 13, which in turn favor IgE class switch or class switch. Rapid immunological reaction occurring within minutes. Rapid immunological reaction occurring within minutes. Minutes after to exposure to allergens include hay fever, asthma, ischemia, food and drug allergy. Food and drug allergy. Most allergens promote a Th2. Let me point that with the mouse so that everybody from the lecture can see where we are reading. Double mediated anaphylactic reaction mediated by IgE attached to mast cells and trigger the granulation of chemical mediators. Most allergens promote Th2, cell that are serial 4, 5, 30 strong favor IgE, class which pathological reaction occurring. Rapid reaction occurring within minutes after to exposure to allergen. This is what all about type 1 hypersensitivity. We used response to foreign antigen, maybe regulatory controlled, resulting to injury. This is the first one, uncontrolled, and promoting the Th1, IL4, IL5, IL30, favoring IgE, rapid immunological reaction occurring within minutes after exposure to allergen includes hay fever, asthma, ischemia, food and drug allergy. Hay fever, asthma, ischemia, food and drug allergy. All of them are the type 1 hypersensitivity uncontrolled type anaphylactic reaction between parentheses. Next, we have a picture explaining the hypersensitivity type 1 anaphylactic reaction. One is urticaria, or has pale red raised itchy bumps. Urticaria, or hives, pale red, raised itchy bumps. Raised itchy bumps, as we can see in the picture. Very visibly, we have the urticaria, hypersensitivity type 1, anaphylactic reaction. IgE has been promoted here, and also the IL4, 5, and 13, favoring IgE. Also, we have the Th2 cells was promoted. Type 2 antibody. Type 2 antibody-mediated cytotoxins. This type of hypersensitivity is caused by antibodies that react with antigens present on cell surface or in extracellular matrix. Let's repeat. This one is caused by antibodies that react with antigens present on cell surface or in the extracellular matrix, present on cell surface or in the extracellular matrix. Type 2, antibody-mediated cytotoxicity. The target cells are red blood cells, and platelets. These are the most critical cells in type 2 reaction or type 2 cytotoxicity. This type of toxicity caused by antibodies that react with antigens present on cell surface from the extracellular matrix. Or the extracellular matrix. and they target WEC, BC, RPC, and plate BC LETs. This type mediated by antibody IgM, IgG. It does not get mediation with IgE, because if it does, then it would be called antibody. It won't be called antibody-mediated cytotoxicity. It will be called anaphylactic reaction, type 1 hypersensitivity, and not antibody-mediated cytotoxicity. This type is related with IgM and IgG. This antigen may be part of cell membrane or circulating antigen, or hapten that attaches to the cell membrane. Part of cell membrane, circulating antigen, or hapten. Let's move to the next slide. We have cell lysis results due to. I think this is a follow-up of the previous slide, which was talking about hypersensitivity type 2, and it's called antibody or antigen antibody-mediated cytotoxicity. We have with this antibody cytotoxicity, mediated antibody-mediated cytotoxicity. We have cell lysis. So cell lysis results due to complement fixation to antigen antibody complex on cell surface, to phagocytosis is enhanced by the antibody, and 3, antibody dependent cellular cytotoxicity. So the cell lysis results due to these 3 important factors. Number 1, complement fixation to antigen antibody complex on cell surface. I don't fully understand the complement fixation to antigen antibody complex on cell surface. Not fully understand this meaning, but we can say that it is a result to cell death or cell lysis. Complement fixation. So the second result of cell lysis is due to phagocytosis, and phagocytosis is enhanced by antibody. Phagocytosis enhanced by antibody. Number 3, antibody-dependent cellular cytotoxicity, ADCC. Antibody-dependent cellular cytotoxicity. Antibody-dependent cellular cytotoxicity. Let's take a rest after this slide, and we'll be back soon, but let's finish this slide first. So first of all, with this slide we have the transfusion and RH incompatibility autoimmune disease. Number one being the transfusion reaction due to ABO incompatibility. The transfusion is a cause of hypersensitivity type 2, or is also called antigen hypersensitivity, as I remember, antibody-mediated cytotoxicity, not antigen, antibody-mediated cytotoxicity. This cytotoxicity is due to or result from transfusion. A transfusion is a reaction or a hypersensitive reaction occurs due to ABO incompatibility. Number two, RH incompatibility, RH, hemolytic disease of the newborn. RH could be negative or could be positive, but this time, if it happens within a newborn, hypersensitivity type 2 will occur. Of course, it won't be type 1 because that would be type 1. We remember this was rashes, stuff like urticaria, hay fever, asthma, ischemia, food and drug allergy. These are all type 1 anaphylactic reaction. The first reaction is RH and transferred to ABO and caused by hemolytic disease. Then 3, we have autoimmune disease. The autoimmune disease, the mechanism of tissue damage is cytotoxic reaction. The mechanism of tissue damage is cytotoxic reaction. Autoimmune disease. We'll mention about an example of autoimmune disease. We have the SLE, autoimmune hemolytic anemia, idiopathic thrombocytopenia, purpura, and myasthenia gravis, nephrotoxic nephritis, Hashimoto's thyroid. Well, sorry about that. We had some difficulties. But we're back to finish off with this slide. This time, hopefully, we won't let it slide. We will not let it slide. And we'll be finishing off with this slide. So, let's start. First, I want to test the voice. Okay, the voice seems to be working fine. Let's go with this slide, slide number 16. Transfusion reaction. This was due to hypersensitivity type 2 and RH incompatibility. Also, there is some autoimmune disease. The mechanism of tissue damage is cytotoxic reaction. We repeat, autoimmune disease. This damage is due to cytotoxic reaction. Example of this autoimmune disease is LA, autoimmune hemolytic anemia, idiopathic thrombocytopenic purpura, and of course myasthenia gravis, nephrotoxic nephritis, and Hashimoto's thyroiditis. We'll be moving on to the next slide now. Four, we have a non-cytotoxic type 2 hypersensitivity. What does that mean is non-cytotoxic type 2 hypersensitivity. Very important one, actually. And it's all about the Graves' disease. Non-cytotoxic type 2 hypersensitivity. The Graves' disease, it is a form of thyroiditis in which antibody are produced against TSH surface receptors. This leads to mimic effect of TSH and stimulate cells to overproduce thyroid hormones. Let's fix the mic. Okay, there we go. Now, a non-cytotoxic type 2 hypersensitivity is Graves' disease, form of thyroiditis in which antibodies are produced against TSH surface receptor. And of course, this leads to a mimic the effect of TSH and stimulate cells to overproduce thyroid hormones. Graves' disease. What is Graves' disease? This is a form of thyroiditis in which antibodies are produced against TSH surface receptor, leading to mimic effect of TSH stimulating cells to overproduce thyroid hormones. 5. Graft rejection cytotoxic reaction. What is a graft rejection cytotoxic reaction? In hypersensitive reaction, the recipient already has formed antibody against the grafts. Graft rejecting cytotoxic reaction. We repeat it. An hyperacute rejection the patient already has performed antibody against the graft. Interesting. Now, Let's go with the drug reaction. Drug reaction includes penicillin, and penicillin may attach as hepatins to RBCs and induce antibodies which are cytotoxic to the cell drug complex, leading to the hemolysis. We can repeat that with the drug reaction of penicillin. The penicillin may attach as heparin to RBCs and induce antibodies which are cytotoxic for the cell drug complex, leading to the hemolysis. Let's repeat that one last time. Penicillin may attach as a heparin to RBCs and induce antibodies which are cytotoxic for the cell drug complex, leading to the hemolysis. The graft rejection cytotoxic reaction. In hyperacute rejection, the recipient already has performed antibody against the graft. This is an hyperacute reaction of graft rejection. And of course, number 4, a non-cytotoxic type 2 hypersensitivity is Graves' disease. Could be question of which one is a non-cytotoxic type 2 hypersensitivity. And the answer is Graves' disease. Graves' disease is a form of thyroiditis in which antibodies are produced against TSH surface receptor. This leads to mimic the effect of TSH and stimulate cells to overproduce thyroid hormones. This is all of Graves' disease. Next, we have this picture showing us type A antigens on red blood cells. Then we have the donated red blood cells with B antigen. The type A antigen on red blood cells of patients. Then we have the donated red blood cells with B antigen, which have a different shape. With transfusion, number 1, complement, then hemoglobin. Number 2, aggregation of complement binding. Number 3, hemolysis with hemoglobin. Of course, this is anti-B antibody sticking to the B red blood cells, causing them to coagulate and agglutinate and cause a hemolysis. Hemoglobin is being produced and the red blood cells type B is getting the anti-B antibody. This was, of course, the slide number 18, and moving on to the next slide, we have another picture explaining the pregnancy. So the pregnancy is a process of giving birth to a child. I think this is pretty obvious. But when it comes to the average difference between the mother and the child, there will be a problem. Giving birth, as we can see here, with the placenta and maternal circulation. The RBCs of the child with Rh antigen. First, pregnancy can be delivered easily. But the mother with the Rh negative, specific B cell combining to the antigen anti-Rh IgM and the plasma cells, as shown in the picture. Then the memory cells coming back to stick to the second pregnancy child, causing him or producing IgGs, adaptive immune system, and the plasma cells attacking the red blood cells of the child. IgG, anti-Rh, AB, crosses placenta and attacks the fetal. Attacks the fetal RBCs, causing erythroblastosis fetalis, which can be deadly. This is a diagram showing the mother treated with Rh O GAM, ROGAM. The B cell with Rh O GAM stick together and prevents the antigen or the antibodies to attack the red blood cells of the child, preventing the B cells activation and more cells formation. Moving to the next slide. Now, in this slide, we have the type 3 immune complex mediated hypersensitivity. I thought there was only two types, but there actually is another type called immune complex mediated hypersensitivity. Repeated, it is immune complex mediated. And it's not anaphylactic and not antigen mediated sensitivity, hypersensitivity. This type is large when a large amount of antigen and antibodies form complexes in blood. We can see this type of antigen, a large amount of antigen and antibody form complexes in the body. Causing coagulation. If not eliminated, can deposit in capillaries or joints and trigger inflammation. PMNS and macrophages bind to immune complexes via FER and phagocytize the complexes. PMNS and macrophages bind to immune complexes via FER, F-E-R, and phagocyte the complexes. But if neutrophils and macrophages are unable to engulf the immune complexes, these cells will degranulate in the area of immune complex, depositing and triggering inflammation. Depositing and triggering inflammation. So hopefully they engulf them pretty much before they do the inflammation, before they trigger it. The neutrophils and the macrophages should be more than enough to engulf the immune complexes. PMNS and macrophage bind to immune complexes. I think this is the type 3 immune complex. Going on, there is type 3 immune complex picture shown off the hypersensitivity reaction of this type of hypersensitivity. Figure 3, when we have the antigen and antibody forming complexes and being engulfed by the neutrophils and macrophages. Very obvious drawing, and that was slide number 21. Take a look at this slide very well while moving to the next slide. This one can cause localized reactions. Localized reactions, meaning inflammations, arthritis, post-streptococcal glomerulonephritis, post-streptococcal glomerulonephritis, skin response to type 3 is referred to as an arthros reaction. Arthros reaction of this type, and it's characterized by local erythema and some induration. Can cause systemic reactions. Acute serum sickness. Acute serum sickness. Of course, we have mentioned this arthritis reaction is caused by local 3 months from individuals, and duration can cause systemic reaction. And 1 second, I want to change the mic. There we go. Now it's much better. I guess. Let's finish off with this, which can cause systemic reaction. The acute serum sickness from large amount of antigen, such as injection of foliin serum. Sickness is usually transient immune complex disease with removal of antigen source. Serum sickness from large amount of antigens such as injection of foliin serum. Serum sickness is usually transient immune complex disease with removal of antigen sources and iopose erythematosus. 1 second while taking a 25-second rest, fixing the mic. Okay. I think we have fixed it already, and we can move on with the process. With the process of learning, pathology, specifically immunology. Now, this one can cause localized reactions. Arthritis, post-streptococcal glomerulonephritis. This one is the type 3 hypersensitivity. Skin response to type 3 is referred to as arthros reaction and is characterized by local arrhythmia and some induration. Can cause systemic reaction, acute serum sickness. Serum sickness from large amount of antigen, such as injection of foliin serum. Serum sickness is usually transient immune complex disease with removal of antigen, source system inputs, and erythematosis. Erythematosis. Moving on to the next slide. In this slide, we have this unfortunate picture showing us the rheumatoid arthritis. The rheumatoid arthritis is an immune complex deposited in the joint caused by inflammation. This one is from type 3 hypersensitivity. The rheumatoid arthritis immune complex deposition in the joint resulting in release of inflammatory chemicals and the joints being to break down and become distorted. Let's move on to the next slide, and hopefully no one gets this hypersensitivity. This one is the systemic lupus erythematosus. Systemic lupus erythematosus. It is different from the previous one, the inflammation of arthritis, rheumatoid. But this one looks like the second hypersensitivity, but this one is actually the third hypersensitivity. It looks more like the first hypersensitivity anaphylactic reaction, but this one is the third one, the third hypersensitivity reaction. Systemic lupus erythematosus. This type of reaction is a hypersensitivity caused by lupus, and auto-antibodies against DNA result in immune complex formation. This type is type 3 hypersensitivity, caused against the DNA and an immune complex formation. Moving on to the next slide. Type 4 delayed or cell-mediated hypersensitivity. This type, the delayed or cell-mediated hypersensitivity, is the fourth type of hypersensitivity. There are many points to indicate with this type, being the inflammation, which could take 24 to 70 hours after contact with certain antigen, is due to action of antigen presenting cell and T cells. The APCs, which are the antigen presenting cells, and of course the T cells, B cells. Delayed reflects the time it takes, or the delay reflects the time it takes for macrophages and T cells to migrate to and proliferate at the site of the antigen. So this is the time takes the macrophage to travel. Takes time, and the time could be between 24 to 72 hours after contact with a certain antigen. Clinical complex or clinical example. In this clinical example, we have type 1 diabetes mellitus, multiple sclerosis, and contact secondary dermatitis. So, first one being the type 1 diabetes mellitus is actually a hypersensitivity delayed reaction type 4. And multiple sclerosis could also be related to type 4 type 4 hypersensitivity. Then we have the contact sensitivity, dermatitis, also a delayed reaction. Moving on to the next slide. We have the essential or the classical example of DTH which is a delayed hypersensitivity reaction, is tuberculin. The tuberculin reaction. Tuberculin reaction is an injection of purified protein derivatives. Tuberculin reaction is an injection of purified protein derivative, tuberculin, beneath the skin. As we can see here, tuberculin is an injection of a purified protein derivative beneath the skin, causes reaction in individual exposed to tuberculosis or tuberculosis vaccine. We talked about this pretty briefly, the classical example of DTH, which is type 4 hypersensitivity, is tuberculin tuberculin reaction. Tuberculin reaction is an injection of purified protein derivative tuberculin beneath the skin causes reaction in individuals exposed to tuberculosis or tuberculosis vaccine. Prolonged DTH reaction against persistent microbes. Prolonged DTH reactions against persistent microbes or other stimuli may result in specific micrological reaction called granulomatous inflammation. Granulomatous inflammation. That one is the prolonged DTH reaction happening against the persistent microbes or other stimuli may result in special morphological reaction called granulomatous inflammation. Granulomatous inflammation. Table 5, we have the classification of hypersensitive reaction according to the gel and compounds. We have type 1, type 2, type 3, and type 4. Alternative name, then associated disease, and mediators. Of course, the mediator with the first hypersensitive reaction is IgG, the atopy, analysis, asthma. These are all from type 1, anaphylaxis. The immediate hypersensitivity, alternative name is immediate hypersensitivity. Number 2 reaction or number 2 hypersensitive reaction is antibody-mediated, associated with the IgG and IgM. This one is autoimmune hemolytic anemia, erythroblastosis fetalis, and good postures disease. These are all type 2 hypersensitivity. The type 3 hypersensitivity is the immune complex mediated hypersensitivity. Immune complex mediated hypersensitivity. This one is a little bit different from the others because it is mediated by the IgG and complement. This one is serum sickness, arthros reaction, and lupus nephritis. Lupus nephritis. The fourth and last hypersensitive reaction is the delayed hypersensitive reaction caused with or happening with or associated with transplant rejection, contact dermatitis, and tuberculosis. It is mediated by T cells, macrophages, and histocytes. Mediated by T cells. Next, we have the types of tissue transplants. Types of tissue transplants. I want to test something. Give me some time. Okay, does it work? Now, types of tissue transplant grafts: autograft, allograft, isograft, and xenograft. First one is autograft, occur in the same individual. Example, skin transplant. Allograft, transfer of organism from one individual to another of the same species. Person to person, example, liver and kidney. Most people do the allograft when transporting their kidneys, and we're talking about transporting the lungs and transporting the kidneys, transporting the liver and other organs of the body from one human to another. Then we have the isograft, which is a graft between members of the same species with identical genetic makeup, identical twins. This is more successful because it happens with very similar genetical traits between two species or two individuals. Then we have the zoonograph, transplant of crown organ from one individual to another of different species. This one might not work because from one individual species to another different individual species. The easiest one to be operate is the isograph. Then one harder one is the allograph. Then the autograph is actually very easy because it's occurs in the same person. Auto, allo, iso, and zono. We will take spend some time before going to the next slide. 5 seconds to contemplate and try to understand this and remember this slide. This picture shows us the autograph from one mouse to the same, showing us the isograph from one mouse to his or to its identical twin. Then we have the autograph from one mouse to another, and of course the zoonograph, which is very hard and very difficult to accomplish. This next subject, we will be talking about immunodeficiencies or immunodeficiency disease. Immunodeficiency diseases may be caused by inherited defects affecting immune system development, primarily, or they may result from secondary effects of the other disease. Example: infection, malnutrition, aging, immunosuppression, and autoimmunity of chemotherapy. The immunodeficiency diseases. Immunodeficiency disease may be caused by inherited defects affecting immune system development primarily, or they may result from secondary defects of other diseases. Example: infection, malnutrition, aging, immunosuppression, autoimmunity, or chemotherapy. These are the immunodeficiency diseases. The primary immune deficiency. Primary immune deficiencies are rare, but most primary immune deficiency diseases are caused by mutation in genes involved in adaptive immune, hormonal, or cellular. Primary immune deficiency. Of course, they are rare, but most primarily immune deficiency diseases are caused by mutation in genes involved in adaptive immune, humoral, or cellular. Cellular or humoral immunity, these are the pure primary immune deficiency. Caused by mutation, of course. I want to try something after this slide. I hope it will work. But you gotta bear with me, so take a rest for 10 seconds after the slide ends. Now, between parentheses, we have lymphadocyte maturation or function. Lymphadocyte maturation or function. Or an innate immunity, including complement proteins and cells such as phagocytes and NK cells. Lymphocyte maturation or function, or an innate immunity, including complement proteins and cells such as phagocytes and NK cells. Okay. Back to studying, but I will take another 5 seconds or 20 seconds of rest. So bear up with me and wait for a while. Okay, we are back. And let's study this primary immunodeficiency are rare, but most importantly, immunodeficiency diseases are caused by mutations in genes involved in adaptive immunity, hormonal or cellular, lymphocyte maturation or function, or innate immunity include component, protein and cell, such as phagocytes and NK cells. Okay. Lymphocytes immunogen functions include innate immunity cells, including complement proteins, and cells such as phagocytes and NK cells. lymphocyte mutations, or innate. I really don't get this second paragraph, but we'll move on. Primary immune deficiency, rare. Of course, they are rare, and immune deficiencies are caused by mutation in genes related to immune hormonal cell. Next slide. We have, of course, some examples of these disorders. Number one being the X-linked agammaglobulinemia. Agammaglobulinemia, okay, agammaglobulinemia, agammaglobulinemia, X-linked globulinemia. Okay. X-linked agammaglobulinemia. Some examples of these disorders are X-linked agammaglobulinemia, failure of pre-B cell to differentiate into B cell. These results absence of gamma globulin in the blood. Second, we have the common variable immunodeficiency characterized by hypogammaglobulinemia. Common variable immunodeficiency characterized by hypogammaglobulinemia, impaired AB responses to infection. Number three, selective IgA deficiency, failure of IgA production. Number four, severe combined immunodeficiency as CID, failure of T cell and B cell maturation. So this one is failure of T and B cell. The third one was the IgA deficiency, failure of IgA. Second one was variable immunodeficiency characterized by hypogammaglobulinemia, impaired of AB, antibody response to infection. And of course, the X-linked agammaglobulinemia, agammaglobulinemia, failure of pre-B cell to differentiate into B cell. Okay. Common variable deficiency, selective IgA deficiency, severe combined immunodeficiency, SCID. Then we have autosomal SCID, failure of T cell development, secondary infection in AB response. X-linked hyper IgM syndrome, failure to produce isotype switched high affinity ABS, IgG, IgA, and IgE mutation in gene encoding. This one is X-linked hyper IgM syndrome, failure to produce isotype switched high affinity ABS. This one is a failure of the mutation in the gene encoding of CD40L. These are all of the examples of this disorder of immunodeficiency. One is X-linked, second is common variable immunodeficiency, selective IgA deficiency, severe combined immunodeficiency, autosomal ACID or SCID, which is severe combined immune deficiency, autosomal severe combined immune deficiency. Then we have X-linked hyper IgM syndrome. Next slide. Slide number 34. We have secondary immune deficiency. Secondary immune deficiency, which may arise as secondary to other diseases or therapies, are much more common than the primary. Much more common than the primary. One second. Okay, we are back. Secondary immune deficiency, which may arise as secondary to other diseases or therapies, are much more common than the primary secondary type may be encountered in patients with cancers, infections, malnutrition, or side effects of immunosuppression, irradiation, or chemotherapy for cancer and other diseases. One example of this type is acquired immune deficiency syndrome, AIDS. We repeat, secondary immune deficiencies, which may rise as secondary to other diseases or other therapies, are more common than the primary secondary type may be encountered in patients with cancers, infections, malnutrition, or side effects of immunosuppression, radiation, chemotherapy is the source of secondary immune deficiency. The one example of this type is acquired immune deficiency, AIDS. This one is secondary immune deficiency. The primary immune deficiency is another type of immunodeficiency. We have the slide 35, RNA virus, HIV, then AIDS, then we have HIV-2, Africa, then we have patients with low CD4 T cells. Then we have virus prevalence in homosexual, promiscuous, homosexual, IV drug users, transfusion, infants born in infected mother, opportunistic infections with pneumocytosis, carini, Candida, albicans and Mycobacterium, avium, etc. Patients with HIV have high incidence of cancers such as Kaposi sarcoma. So all these are the secondary immunodeficiency diseases. HIV to patient with OCI, CD4 T cells, CD4s being the T helper cells, TH. Virus prevalence in homosexuals, IV drug users, transfusion and support with and infections with pneumonitis, etc. Avium, patient with HIV and have incidence of cancer such as Kaposi sarcoma. We have these pictures showing all the secondary immunodeficiency diseases. Lipid membrane, matrix, RNA caspid, reverse transcripts, GP41, transmembrane, glycoprotein, then with GP120, docking oligosapoprotein. All of these are some reasons to exploit the cell and make it more vulnerable to diseases, one of which is secondary immunodeficiency diseases. Thank you for listening. This was a studying for pathology, immunodeficiency, and immunologic hypersensitivity. Hope you enjoyed, and we'll catch you next time on this amazing and cool way of education and understanding each subject of this matter. Thank you all for listening, and goodbye.